So first let's see how glycine metabolism takes place. Glycine is an amino acid that is widely used for the synthesis of protein. It's a non-essential amino acid and it's also glucogenic, remember. So one way of glycine synthesis is from serine. Okay, as you can see here, from serine, after removal of this group in the presence of vitamin B6 cofactor, hydroxymethyl transferase can convert them into glycine. Second way how glycine can be synthesized is with the help of the amino acid called threonine. The amino acid threonine can help also to synthesize glycine. Third way the glycines can be synthesized is the help of betaine. Okay, betaine. From betaine it can be used also. So what happens? How is glycine degraded? Glycine can be degraded by the help of the enzyme amino acid oxidase. See, if we have excess glycine, it can lead to a condition called hyperoxaluria, which can ultimately cause kidney stones or renal stones. Okay, so excessive glycine degradation also may lead to excessive oxalate production, which may cause these renal stones here. So glycine is basically degraded or broken down and converted into glycosylate, as you can see here, oxalate, and so on. Okay, so glycosylate basically will help to do so. Function of glycine, see here, it helps in the formation of proteins, hormones, and enzymes, also heme group of hemoglobin, in synthesis of purines, creatinine, glutathione, and so on. Okay, so how glutathione is formed from glycine? You can see here the second one, formation of glutathione. We see that special function of glycine includes formation of glutathione. So when glutamate basically along with cysteine can be converted into glutamyl cysteine and glycine, which ultimately converted into glutathione by the help of the enzyme MSH synthetase. Glycine also plays roles in the formation of creatinine. Not creatine, creatinine, it's creatine. Okay. Glycines in the kidneys can be acted upon by an enzyme called arginine glycine trans amidinase and can be converted into ornithine, which in the liver is ultimately can be converted into our ketone phosphate and then creatinine. See here, the enzymes that is involved in the conversion of ketone phosphate to creatinine is CPK, ketone phosphorokinase. Moving on. Number 2, serine. The amino acid is non-essential and again glucogenic. Now this one can be synthesized from the glycine. Okay, so it's reversible reaction. The one that we've seen, it can be reversible. So we can generate serine by the help of glycines. Okay, it can also act in the intermediate of glycolysis. How is serine degraded? It can be converted into glycine and ultimately can be eliminated as a form of carbon dioxide and ammonia, the major way. The second way is that it can be converted into pyruvate by the help of serine dehydratase. The pyruvate then can enter TCA cycle or can be converted into alanine or can be used for synthesis of glucose. Serine is important because it plays roles in the synthesis of phosphoprotein, purines and pyrimidine, sphingosine, choline, cysteine, etc. So serine plays roles, as you can see here, in the synthesis of glycine, phosphoprotein, purine and pyrimidine, and so on. Okay. Next one, sulfur containing amino acids. Which are our sulfur containing amino acids? These are methionine. There's a spelling mistake. It will be cysteine and cysteine. So methionine can be converted into cysteine. Okay, from dietary protein by the process of transmethylation and transsulfuration. You can see here methionine can be first go through the process of transmethylation and be converted into S-adenosyl methionine. Okay, now if we don't have the enzyme cysteine synthetase, then excessive amount of methionine ultimately can result in a homocystinuria, a condition also. Second one, how do we go through the metabolism of cysteine and cysteine? They are interconvertible, that is, one can be converted from one to another easily. So as you can see here, they both can be synthesized by methionine and serine, and ultimately both can be interconvertible, cysteine, as you can see. So cysteine can be converted into cysteine, can be pyruvate, glutathione, and so on. Methionine can give rise to homocysteine. Excessive amount can cause a condition called homocystinuria. Homocysteine then can convert into cysteine, and so on. 4. Aromatic amino acids includes phenylalanine, for example, number 1, okay, which is a glucogenic and ketogenic amino acids. It also includes tyrosine. These two are basically our aromatic amino acids. So phenylalanine ultimately is converted into tyrosine by phenylalanine hydroxylase. Okay, tyrosine then can convert into hydroxyphenyl pyruvate and ultimately can convert into homogenistate and malonyloacetate, then to fumarate acetate, which ultimately will convert them into fumarate. So if fumarate can enter the Krebs cycle and help in gluconeogenesis, or can be also converted into acetoacetate, which can help us in the production of lipids. So as it has both the properties, glucogenic and ketogenic. Second one, tyrosine, is a precursor of DOPA, or 3,4-dihydroxylphenylalanine. The tyrosine can be converted into DOPA, which is a very important transmitter, neurotransmitter you can call it. Okay, as you can see here, our tyrosine that we have can be easily converted into our dopamine by several mechanisms, which can be converted into norepinephrine and so on. Thyroxine formation. Okay, so these hormones, these amino acids also may play a role in the formation of hormone called thyroxine, or thyroid hormone, which is very, very important. Okay, the third aromatic one is tryptophan. Tryptophan is both essential, glucogenic, and ketogenic. Tryptophan can be converted into a compound called kynurenine, which can be ultimately converted into hydroxykynurenine, and later on can enter both the glucose synthesis mechanism via the lipid mechanism via acetoacetyl coenzyme, or can also enter and generate NAD and NADPH. The other way by which the tryptophan can be metabolized is called the serotonin pathway. So tryptophan can play a role in the synthesis of a neurotransmitter called serotonin. Okay. So for what happens here, tryptophan first gets converted into 5-hydroxyltryptophan, then into 5-hydroxyltryptoamine, the serotonin that we have, which also has the ability to convert into melatonin, a pigment, melatonin and hormone, which is very important as well. Serotonin can convert into O-methyl, by the help of O-methyl transferase, can convert into melatonin. See here, melatonin formation. This hormone is found in the pineal body in the brain of a man. It can be formed by serotonin with help of methylation and acetylation. Okay, it's a very important hormone. It can help in regulation of our gonads and adrenal function. It helps us to maintain our integrity of cells, and it has an antioxidant property. It also enhances our body's defense against infections such as AIDS, and it also reduces the risk of cancer and heart diseases. Branch chain amino acids, leucine, isoleucine, and valine. These comes under the group of branch chain amino acids. These amino acids are mainly taken up by muscles. After you have taken a meal, specifically, okay, they are taken up by the usually skeletal muscle, but after a meal, it can be oxidized in the skeletal muscles. They are used in the brain, these amino acids. So what happens basically, leucine can ultimately leads to production of acetyl coenzyme and acetylacetate. Isoleucine can form succinyl coenzyme and acetyl coenzyme. Same goes for valine. It can form succinyl coenzyme and carbon dioxide. Number 6 is our basic amino acids. This includes number 1, histidine, number 2, arginine, and number 3, lysine. Histidine is known also as a glucogenic amino acids. Together with our alanine amino acid, it can form something called carnosine and anserine. Okay, these are buffer of a pH, especially in cases of skeletal muscles. They can activate our myosin ATP-ase and also can remove copper or enhance copper uptake. Histidine is also a source of one carbon atom. See here, it can also form histamine, which act as a messenger that mediates allergic and inflammatory reaction, gastric acid secretion, and neurotransmission of the brain. Number 2 is our arginine, non-essential amino acid, glucogenic. It helps in the formation of creatinine, polyamines, and nitric oxides. Okay, see here, the arginine can be converted into citrulline also. We have seen in the urea cycle here. The citrulline here can ultimately leads to nitrogen oxide, which acts as a free radical and helps us to release our smooth muscles, prevents aggregation of platelets, and also acts as a neurotransmitter in brain. Number 3, lysine, essential ketogenic. It plays roles in the formation of histone, hydroxylysine, and carnitine. Okay, number 7, acidic amino acid, glutamic acid. It's a non-essential amino acids, okay, and glucogenic. It helps in the formation of glutathione and proline. Also, you see here, glutamic acid can be converted into glutamine, which acts as a storage form of ammonia, and also plays roles with GABA neurotransmitter in the brain. Number 2 comes under this acidic amino acid is aspartic acid, non-essential and glucogenic. It can lead to generation of arginosuccinate in urea cycle. It can form alanine by decarboxylation and oxalate and glucose by the TCA cycle. Some of the amino acids you have seen act as a neurotransmitter of the brain. So arginine acts as a precursor of nitrogen oxide, tryptophan for serotonin, histidine, histamine, phenylalanine, dopa, dopamine, norepinephrine and epinephrine, glutamic acid, GABA. Okay, so you should know this. This is a high yield content amino acids which are precursor of neurotransmitter. You must know these amino acids. Okay, in detail you can find also over here serine and choline act as acetylcholine and so on. You must know these in details. Now, during this whole process of amino acid metabolism, errors may take place. In some patients, there might be errors and they might have some defects. So what are their clinical significance on those individuals who have some kind of defects or errors in amino acid metabolism? If you look into this chart, just to give you a brief idea, you don't need to know in details because you will study this in medicine. You know what kind of defects can lead to what kind of conditions. For example, albinism. Okay. Defective process is what? Melanin synthesis from tyrosine. Enzymes, tyrosine 3 monooxygenase. Symptoms, lack of pigmentation. Alkaptonuria, tyrosine degradation processes, there is defect. Arginemia, urea synthesis defect. Arginosuccinexidemia, urea synthesis defect. Carbonyl phosphate 1 synthesis deficiency, urea synthesis defect. Homocystinuria, methionine degradation defect. Maple syrup urine disease, problem with isoleucine, leucine and valine degradation. Phenylketonuria, conversion of phenylalanine to tyrosine. This process is defect. So there is high amount of phenylalanine. Okay, patients will have vomiting, mental retardation, one of the common one. Okay, phenylketonuria. So these are some of the medical conditions that can develop if we have any kind of defect in protein metabolism or amino acid metabolism, or if there is any kind of problem. So for this lecture, okay, the main thing that you should know is the types of amino acids that we have, their characteristics. You should be able to know that how the proteins are digested, different phases, gastric phase, pancreatic phases, and intestinal phases. You should be knowing certain definitions, the certain terms such as nitrogen balance, and as well as bioavailability or biological value. You should be able to know what happens with the amino acid, what are the phases of the amino acids, okay, what are the process in details. You should also know, for example, deamination, transamination, and so on, okay, how the amino acids are metabolized. Okay, you should be able to know also the metabolism of ammonia, what happens to the body's ammonia, how they are being removed. Okay, this metabolism of ammonia is very important content, you should be knowing this. What happens if we can't remove it, leads to ammonia poisoning or ammonia intoxication. And the most important content is urea cycle. You should be able to know the 5 stages of urea cycles, the enzymes involved in there, okay, what is going on in the process in details. You should be knowing about this. How the urea cycle is regulated also is very important. What will happen if the urea cycle has a defect? Okay, you should be able to know that. And lastly, about the metabolism of individual amino acids is just to give you a brief idea. You don't need to know in details what happens to individual amino acids, just to have a reading and have a brief idea about the fate of the amino acids individually. Okay, so this is all the contents that we have regarding our protein catabolism. Thank you, and if you have any question, you can always ask if any confusion or question you have. So let's check out for the next time. Thank you very much.